Okay, we're on slide 5 on cell reproduction. Okay, so we're going to talk about the eukaryotic cells. So, movements of substance over long distance is slow and difficult. So, cells remain small to maintain efficiency. When a cell reaches its size limit, it will either stop growing or it divides. Cell division keeps cells from getting too large. It is also the way cells reproduce. A cell's cycle of growing and dividing is called a cell cycle. Okay, so there are two reasons why a cell divides. Okay, okay, all right, so it says right here. Okay, this part right here we're going to read here. There are three main stages. During interphase, there's one, you're going to type in the word interphase. The cell grows and carries out cellular functions and copies its DNA. Interphase is followed by mitosis. No. And mitosis is the period when the nucleus divides. So here's the first stage, interphase. Here's the second one, mitosis. And that's when the nucleus divides. And then here's the last one, cytokinesis. Cytokinesis. This follows, that goes here, that follows mitosis and is the stage when cytoplasm divides and then two cells are created. Okay, so those are your three stages: interphase, mitosis, cytokinesis. And it tells you a description of each one that you're going to type in here. Okay, in your own words. Okay. What happens during interphase? During interphase, which takes up most of the cell cycle, the cell grows, performs normal cell functions, and copies its DNA in preparation for cell division. Interphase is divided into three stages: gap 1, synthesis, and gap 2. Okay, so here we go, right? As soon as the cell divides, it enters the G1 stage where it grows and performs normal cell functions and prepares to copy its DNA. Okay, I'm going to escape for a second because you can't really see the whole slide. So you're going to type in here what's happening and at what stage. Okay, so this is your interphase, and then G1 is the stage where the cell is growing and it's performing its normal functions and it's preparing to copy its DNA. Okay, and then during the S stage, the cell copies its DNA. Chromosomes are the structures in the nucleus that contain the DNA, the genetic material that's passed from generation to generation of cells. The DNA in the chromosome is tightly wound. Chromatin is relaxed and unwound to form of DNAs in the nucleus. Okay, and finally, the G2 stage is the period when the cell prepares for division in its nucleus. When preparations are complete, the cell enters mitosis, followed by cytokinesis, and the end result of the cell turning into its original cell into two identical daughter cells. Okay, so interphase, and then each of these are your G1, your S, and your G2. Okay, it says drag the shape to the stage of interphase where DNA is copied. So you're going to drag this. Where is DNA copied? And it tells you in here. Okay, easy. The cell cycle in a eukaryotic cell is controlled by a combination of two substances that signals the cellular reproduction process. Proteins called cyclin bind to enzymes called cyclin-dependent kinesis. In the stages of interphase and mitosis to trigger the various activities that take place in the cell cycle, as shown below. So tell me what the two substances are. It says right here. And according to the figure, in which phase do signals prepare? So you're going to look here, and according to the figure here, how many checkpoints are there, which is really cool. And you're going to look here. And if you watched yesterday's video, the Amoeba sisters talk about these checkpoints that a cell goes through, whether or not they pass. If they don't pass those checkpoints, then the cell is not able to reproduce and make new cells, because these checkpoints are saying, oh, I don't know, you're not looking like you're such a good cell. But I'm sure you learned that yesterday when you watched the Amoeba sisters. And then finally on 8, cells use different combinations of cyclin or CDK to control activities at different stages in the cell cycle. For instance, in the G1 stage, one cyclin CDK combination signals the start of a cell cycle, while another combination signals the DNA replication, protein synthesis, nuclear division, and at the end of the cell cycle. The cell cycle has a built-in quality control checkpoints that monitor the cell's cycle and can stop if something goes wrong. For instance, near the end of the G1 stage of the interphase, the cell monitors the DNA for damage and can stop the cycle before it enters the S stage if something is wrong. So these are those checkpoints, right? The cell is checking and being like, no, there's something wrong with the DNA. There are other quality control checkpoints during the S stage and after the DNA duplication in the G2 stage before entering mitosis. During mitosis, the cell checks the spindles, the fibers, before it undergoes cytokinesis. If the cell detects a failure, the cell cycle stops. Okay, so these quality controls are those checkpoints that the cell does before it replicates. It checks it multiple times. Okay. So you can answer these questions based upon the text right here. Okay, and that's all you're going to do today. Okay, and then we're going to go into these. This is so cool. On Monday. When we're at school and I don't have to show you any more videos. Well, maybe I like the Amoeba sisters. Okay, so you're doing 5 through 8 today.